Assalamualaikum warahmatullahi wabarakatuh. My name is Cik Nur Izzah Izzati, and today my team members and I are going to do the demonstration of the lab DNA extraction. Okay, the most important thing in this experiment or in this process of DNA extraction is the pipetting technique. Okay, this is because as you can see, this is the culture of bacteria where we are going to extract the DNA from this culture of bacteria using a pipette by a pipetting technique. Okay, in this experiment, in this part, it is important for us to make sure that there is no contamination occurs. So it is more recommended to carry out this experiment in a laminar flow or near the fire. Okay. The first thing that we are going to do, we are going to pipette in 1.5 milliliter of culture of bacteria, and we are going to pipette it out into a centrifuge tube. Okay, at this part or at this step, make sure that we pipette it until the end, we press it until the end to make sure that there will be no more culture of bacteria left in the pipette and everything will go into the centrifuge tube. Okay, for the next step is we are going to proceed with the centrifugation process. As you can see here, this is the tube that we have filled it with the culture bacteria. Okay, so this centrifugation process, we will use the centrifugation machine and we will put it into the position of opposite for both of the tubes. Okay, we position it opposite for both of the tube to balance the tube. Okay. In this process, the machine will spin the tube at a very fast, which is for 1 minute. We will put it at 12,000 RPM. Okay, as you can see here, this is our centrifuge tube that contain our culture of bacteria that have gone the centrifugation process. Okay, you can see here. Okay, I will zoom in. Okay, there's two layer of our culture of bacteria, where the upper part, which is the liquid, is the supernatant, we call it supernatant, and at the below is the cell, which is we call it pellet. Okay, so in this experiment we need to use only the cell, so we are going to remove the water. So you just pour the excess water and make sure that there is no more water left. There is no more excess water left. We dry it out by using this tissue paper. Okay, so you can see here, there is no more upper layer and we only left with the cell that we are going to use in this experiment.
After discarding the supernatant, only the pellet is left in the microtube, which is the bacterial cell. Using a yellow micropipette and yellow micropipette tips, add 100 microliter of lysis buffer into the microtube and resuspense the microtube using a vortex. Lysis buffer helps to break down the sour of the cell. Vortexing the micropipette will help to mix the pellet and the lysis buffer better. The pellet will be gone after vortexing, which indicate that the pellet has been mixed up well with the lysis buffer. After vortexing, add 20 microliter of proteinase K into the microtube and incubate it in water bath. Incubation should take for 15 minutes at 55 degrees Celsius. For every 5 minutes, we need to flip the microtube to ensure a proper mixing. the solution is not clear, further the incubation for another 30 minutes.
So the next step, 20 microliter of RNSA, this is RNSA, will be added into the bacteria. The function of RNSA is to degrade the RNA because we only need the DNA. Now we pipette out 20 microliters of RNSA. This one. Then transfer to bacteria. You may vortex it to mix all the solution. The mixture will be incubate for 2 minutes. So the next step, 500 microliter of binding buffer will be added into the bacteria. The function of adding binding buffer is to bind the DNA to the white area of spin column. Now we pipette 500 microliters of binding buffer. Okay. Transfer to bacteria. This is the spin column. It comes with 2 parts. The upper part is called spin column while the lower part is called collection tube. In the spin column, we can see there is white area over here. This is where the DNA will be trapped during centrifugation process. Therefore, these 2 parts need to combine it all together and centrifuge it. After add lysis buffer, proteinase K, RNSA, binding buffer and cell, we will transfer everything inside spin column. So, make sure to pour everything inside spin column. Then, after transfer all the cell mixture, centrifuge it for 30 seconds at 12,000 G to make sure every solution is pushed out from the spin column to collection tube.
So for the binding buffer centrifugation, we're going to take all the solutions that has been poured into this spin column and put the spin column into the micro centrifugation machine and let it spin for 30 seconds at 12,000 RPM. So next is the cleaning and washing buffer step. So after the centrifugation that we had done in the binding buffer centrifugations, we're going to take out the spin column from the micro centrifugation machine. So everything will be flowing out to the collection tube. So we're going to take out the collection tube and then discard all the flow through into the here. So in this step, we're going to include 2 different solutions, which are the clean buffer and also the wash buffer. So the step for both solutions are just the same, but this one we're going to do it 1 time and this one we're going to do it for 2 times. Okay, I'm going to show the step that we are going to do. So the first one is we're going to take 500 microliter of the clean buffer and put it into the spin column. And then we're going to put the spin column into the microcentrifugation machine and let it spin for 30 seconds at 12,000 G. For 30 seconds, we're going to take out the spin column again, and then we're going to take out the collection tube and discard all the flow through again. So the step that I've done with the clean buffer, I'm going to repeat it again but using the wash buffer. So using the wash buffer, we're going to do it twice, but I'm just going to do it 1 time, so just to show it. So the first one is to take again 500 microliter of wash buffer, put in the spin column, okay, and then put the spin column in the microcentrifugation machine and let it spin for 30 seconds at 12,000 G. So the next step is the spinning empty column. So in this step, we're going to take this empty spin column and put it into the microcentrifugation machine for 1 minute at 12,000 RPM. So the purpose of redoing this is to make sure that we remove all the excessive ethanol that are trapped in the spin column. So the last step is the elution step. So after 1 minute of centrifugation of empty spin column, so we're going to take out the spin column and discard the spin column. The reason we discard the spin column because the DNA with the elution buffer has eluted into the collection tube, and now we have our DNA. Then the DNA can be kept at -20 degrees Celsius until to be used. Thank you, that's all from us.